Good morning, and thank you for joining this morning's OSHA Grand Rounds. It's our pleasure to be joined today by Dr. Joseph Loscalzo, who is the Hersey Professor of Theory and Practice of Medicine at Harvard Medical School and Chairman of the Department of Medicine and Physician-in-Chief, and Semmelweis Distinguished Chair in Medicine at Brigham and Women's Hospital. He's a summa cum laude graduate of University of Pennsylvania, where he obtained his MD and PhD in biochemistry. And he's the author of over 1,100 articles, 51 books, and 32 patents. He's internationally recognized for his work on vascular biology of nitric oxide, redox biology, systems pathology, and network medicine. And he's received many, many amazing rewards. And we're so happy to have him. Dr. Loscalzo will be joining us in a few minutes. I just wanted to touch on a couple quick points before we start the lecture. If you're requesting CME credit this morning, please email the instructions and request instructions in the chat. Please post any questions in the Q&A. We're going to have a short Q&A session at the end of the talk. And with that, I'm going to invite Dr. Loscalzo to give us the lecture.
Thank you very much, Catherine. It's a pleasure to be here and to talk a little bit with you about food as an innate drug source and how we might use network strategies that I'll describe for you to develop compounds in foods as potential drug therapies. My disclosures are listed here. And there are 4 parts to the talk. I'll talk a little bit about contemporary drug development and its limitations, then briefly touch on this notion of network medicine, molecular interaction networks, and their relationship to disease, and then use that same paradigm as a way to identify potential drug targets for drug development, and then turn to food biochemicals, nutritional biochemicals as potential drugs and how they might be explored, with a couple of recent examples. Now, if you look at the number of approved drugs, FDA-approved drugs since the early 90s, you'll note, as people in the pharmaceutical industry do, that the numbers really haven't changed very much, a little bit greater approval rates in the last few years, but still the number of new medical entities, or NMEs, these are new structural products, have rarely exceeded 40. And asking anyone in the pharmaceutical industry as to why productivity is so limited despite extraordinary advances in structural biology, their arguments are typically that the regulatory environment is too stringent. There's an increasing need to explore novel targets, which is a corollary to the first point, as is the fact that easy targets have been exhausted, with increasing attrition rates for developing drugs in early phase development. But I would say the major problem is the intrinsically flawed reductionist approach to drug development. That is the need to identify a single drug target with a single magic bullet that affects that target. And the person for whom we need to, or to whom we need to make note, who's established this approach to drug development is Ehrlich. You remember that Ehrlich was a dye chemist in the late 19th century in Germany, and he'd made a series of observations early on in the tissue selectivity of dyes. He noted that, and in fact, some of this differential staining is certainly used even today in histology laboratories and clinical pathology, as you're all aware. But as he thought about what accounted for this differential uptake of different structurally featured dyes, he developed what was known as a side chain theory, arguing that there were receptors without knowledge of their structural components that were avid for certain dyes and that were differentially expressed in tissue. So this notion of receptors for dyes expanded to include the subsequent notion that there were also receptors for specific small molecules that could benefit patients with diseases. And as he published in 1910, humbly describing preparation 606 under his name, he was able to show that an arsenical pulled from his shelf was effective in killing the spirochete. So this was the first low throughput drug screen. He basically simply looked empirically at a whole host of small molecules that he had access to and tested in a straightforward assay, in this case, spiroketocidal activity. So those principles, the notion of selectivity that was receptor dependent, the notion of a single target that could serve as the basis for an effective therapy, the notion of a high throughput screen, the notion of structure-activity relationships, they all evolved from Ehrlich's initial observations. He really connected chemistry with biology and medicine for the first time, and in this sense could be viewed as the first chemical biologist or medicinal chemist. But as was the approach in the 19th and certainly well into throughout the 20th century, his strategy was a reductionist strategy. And I should say that this magic bullet principle really held sway and continues to in pharmaceutical companies, in the regulatory agencies that govern drug development, always asking the question, you'd like to have this drug approved, what's its target? In departments of medicine in the NIH, their structures are all based on this notion of single organs, single diseases, single therapies. Obviously, I'm being a bit hyperbolic here for purposes to be provocative, but the principles are largely, continue largely driving drug development. Now, contemporary clinical trial design using this approach, single drugs, single targets, has been successful to be sure, but not as successful as one might like to believe. For example, the average clinical response rates of all FDA-approved drugs is about 25%. And of the drugs that were approved in 2018, when I looked at these numbers, 42% were based on a single pivotal trial, not even a confirmatory trial. And let me make the point about the inadequacy of current drug efficacy. In this slide, which was adapted from Schurk's work, it was provided by Harold Schmidt for me. For every individual they help, shown in green, the 10 highest selling drugs in the US failed to help between 3 and 24 individuals. It's a pretty striking image. Now, hold that thought and let me turn to another principle I want to share with you, and that is that drugs have many targets. They're very promiscuous. That even though we like to think that there is a single drug for a single binding site for a single target that governs the phenotype and its response, as it turns out, bioinformatic analysis, as shown here from the work of Chartier, shows that if you ask the question, how often does a binding site for a specific drug recur in the human proteome for those proteins for which tertiary structures are known, it's quite high. About 32 targets per drug on average. This is, as you can see, the distribution is long-tailed, and most drugs have lots of targets. Very few have single targets. And that, of course, argues for two important points. One is it explains the adverse off-target effects that we deal with, especially once drugs that have been approved become more widely used and rare events pop up. And the second is it offers an opportunity. Can these drugs be repurposed in order to explore potential alternate uses, as they were developed in the conventional industry paradigm by identifying the disease of interest first, and then the pathway of interest second, and then screening proprietary libraries of drugs third. So this analysis provides a caution, but also an interesting opportunity. So that sets the stage for contemporary drug development principles and their limitations. Now let me briefly now turn to a little bit of a summary of the notion of network medicine and how we can try to marry network approaches to drug discovery. And I apologize a bit for this this long background, but it's necessary to understand the discussion on food biochemicals in the last third of the talk or so. So in 2011, my colleague Laszlo Barabasi, who's a network scientist, a physicist, and close collaborator now for many years, published a concept piece in which we asked the question, or posed the hypothesis, that if one were to ascertain all of the feasible physical interactions between proteins in a cell or tissue, and had that comprehensive network of protein-protein interactions in hand, could one explore that network for subnetworks that govern disease? And we hypothesized that gene products, proteins that govern disease, cluster into subnetworks shown in color here in this interactome, this comprehensive protein-protein interaction network. And exploring those subnetworks should give insight into mechanisms that are specific to those diseases, and equally important, give insight into potential targets that could be explored as possible therapeutic avenues for those diseases. Now, the organizational principles that bear on this hypothesis are listed here. These are themselves described as hypotheses. First is the local hypothesis that proteins involved in the same disease tend to interact. Shouldn't be surprising. Then the disease module or subnetwork hypothesis that proteins involved in the same disease tend to cluster in connected subnetworks. Next, we have the functional coherence hypothesis that proteins in a disease module are often involved in the same biological process, hence the same pathways of interacting proteins. And the last is that the shared components hypothesis that related diseases tend to be located in the same region in the interactome, the same neighborhood, from which unrelated diseases are cleanly separated. And over the last 10 years, we've published a number of papers that support these hypotheses, or this central hypothesis that disease determinants, or proteins governing disease phenotype, do cluster in the interactome, that is the comprehensive network of interacting proteins as we can ascertain them. And here's the first paper, key paper, that highlights this point that we published now 6 years ago or so, in which we curated a host of experimentally validated protein-protein interactions in the human cell, including, importantly, Mark Vidal's seminal work here at the Farber, who has spent the last 20 years putting together protein interaction networks using a variety of approaches, including yeast to hybrid screens. So we've taken Mark's network, our own determinants of protein-protein interactions, a host of bioinformatically available open access networks, and built this interactome. And at the time that we published this paper, it comprised over 13,000 nodes or proteins and over 140,000 edges or interactions between those proteins. This fuzzball in the background is an actual depiction of the human protein-protein interaction network that we knew about in 2015. Notice the ratio of interactions to proteins. It's a little more than 10 to 1. You recall from your first courses in biochemistry when the question was asked as to why proteins are as big as they are. The professor would typically say, because the structural requirements of active sites of enzymes or binding sites of binding proteins are so precise that it requires this large matrix, if you will, of protein infrastructure to maintain it. But that's not true. We know now that far smaller structures can be active catalysts as efficient as enzymes, and certainly far smaller structures can bind to ligands competitively with proteins. So this work, this analysis really suggests to me that the reason proteins are as big as they are is because they interact with so many other proteins on average in the densely packed environment of the cytosol, where there's about a third volume percent of proteins, excluding water significantly, very close together, interacting avidly. So in this gamish, in this protein-rich network in the cell, one can, in fact, identify subnetworks shown schematically, drawn on top of this interactome, for diseases like multiple sclerosis, rheumatoid arthritis, and peroxisomal disorders, shown here for illustration. And as you note, multiple sclerosis and rheumatoid arthritis overlap, as you might predict they might, given that they are both immunologically mediated diseases. And of the 299 diseases for which there were adequate numbers of genes or proteins, adequate meaning 20 or more in the literature, we were able to show that about 85% of them had disease modules that were discrete, subnetworks within the interactome. Now, how do we derive a disease module? Here's an example from the work of Rusheng Wang. This is for cerebrovascular disease. We first ascertained the disease network components, the seed proteins. These are shown in blue, and they come from reductionist experiments or from unbiased screens, gene expression screens or proteomic screens. Then one maps these seed proteins to the interactome. And this is where the benefit of the interactome truly shines, that these mapping these seed proteins allows one to identify connector proteins shown in gray that weren't previously known to be associated with the disease, but provide the links, the missing links among the disease-associated proteins that then lead to the depiction and detection of a disease module. So that disease module serves as the basis for our explorations into drug treatments for diseases. And now let's turn to that part of the talk with this background. Now, from a network perspective, one recognizes quickly that a conventional strategy for drug target identification rarely involves a sort of global or holistic sense of where that target sits. Yes, it's true that for high throughput screening, typically some pathway is analyzed that appears to be related to the function of that potential drug target. But it's generally analyzed out of context. And given how richly connected many proteins are within the cell, it is rarely the case that all possible interactions are explored. Many of them are not or have not been recognized until recently through this kind of network analysis. So we're missing opportunities for exploring drug target interactions and their broadest consequences for disease phenotypes. Another interesting principle that comes from the network-based drug target identification strategy is the principle of drug repurposing, rational drug repurposing or repositioning. And that's illustrated here on the right. Let's say, for example, that one has a disease module shown in this pale blue oval and its network protein components, adjacent to which is a drug target for a disease that's unrelated or believed to have been unrelated to this disease of interest. The question one can ask is, could the drug that has been approved for the treatment of this disease, could it have some effect on this new disease of interest, given the proximity of its target, only 2 interactions away from the disease module of interest? This short path notion of a target to a disease module of interest. So to test this hypothesis, we put together a group of really talented folks who explored this question from the perspective of big data sets, from an administrative database to administrative databases, from the perspective of complex molecular interaction networks, and from the conventional wet lab experimental basis. And let me describe what was done here. This is work that was led by Fei-Sheng Cheng, who was a postdoc at the time and is now at Cleveland Clinic. We first asked the question, Fei-Sheng asked the question, let's consider the disease module for coronary artery disease, which is obscured a bit by this box, but it's sitting in the background here. Now, within that disease module are drug targets for drugs that are unrelated to coronary artery disease. Let's explore some of those targets and ask the question, could the use of drugs for those other diseases influence the incidence of new cardiovascular events in patients being treated for those other diseases? Now, to perform this kind of bioinformatic analysis properly, one needed drugs whose targets for other diseases sit within or very near to the disease module of interest, the coronary artery disease module, and a comparator used to treat the same disease as the drug of interest, but whose target is quite remote from the coronary disease module. And we looked at several pairs of drugs that were defined in this way, and I'll focus just on two. Carbamazepine versus levetiracetam. These are used to treat, as you know, seizure disorders. And hydroxychloroquine versus leflunomide, used to treat autoimmune diseases like RA and lupus. Carbamazepine and hydroxychloroquine have targets that are in or near the coronary artery disease module, whereas levetiracetam and leflunomide do not. Now, the databases we turned to were analyzed by our colleagues Sebastian Schneeweiss and Rishi Desai in the pharmacoepidemiology division of the department. They used the TrueVen and Optum data sets that together comprise over 200 million individuals. And these individuals, one can actually monitor fairly precisely when new drugs are initiated, whether their prescriptions are renewed, how long they're treated for, and the incidence of new clinical events. In this case, we were asking about the incidence of coronary artery disease-related events during treatment, beginning about 3 months after treatment, out to, I think it was 3 years. And what they found, I apologize for how busy this slide is that carbamazepine increased the risk of an incident coronary artery disease event compared to levetiracetam, whereas hydroxychloroquine decreased the risk. Given the huge number of patients individuals in these administrative databases, you can see how large the cells were within which we can make these comparisons with reasonable confidence, recognizing the limitations of any administrative database. So we then turned, in this case, Diane Handy in our research lab, turned to an exploration of potential mechanisms by which hydroxychloroquine could protect against coronary artery disease. And exploring the disease module, she identified two key targets for hydroxychloroquine, TLR7 and 9. These receptors signal to activate a host of downstream processes that enhance the inflammatory response, in this case in the endothelium. And she specifically looked at the expression of an inflammatory adhesion molecule, VCAM, the release of a cytokine interleukin-1 beta, and the suppression of a protective enzyme, endothelial nitric oxide synthase, as a result of TNF stimulation in the absence or presence of increasing doses of hydroxychloroquine. And what her results showed is that, in fact, the hydroxychloroquine could attenuate, not completely reverse, but attenuate the adverse effects, the inflammatory effects of TNF, and in this way may, in part, account for its beneficial effects in limiting the incidence of new cardiovascular events. Now, we've gone on to look at drug repurposing in other settings. This is more recent work by Jun-Sop Song and Jane Leopold, in which they looked at molecules or proteins within the interactome that govern vascular calcification. And within that calcification endophenotype module, they identified a variety of drug targets and then tested the effects of these drugs on calcification. And there were two groups, 3 drugs that turned out to be interesting. One is everolimus and temsirolimus, both of which are mTOR inhibitors, and the other is pomalidomide, which is a thalidomide congener that affects a host of signaling pathways that govern calcium deposition in the vasculature. And Jane showed in this case that all 3 of these significantly suppressed, at least in an in vitro assay, calcification pathways. The plan would be that such drugs could be tested in patients who are at great risk for calcification. Now, one more example before we turn to food, this is an example of recent work that we just published now about a month or so ago. Actually, it was earlier in April in PNAS, and then subsequent work, which is under review but published in bioRxiv, that I'll show you as well. Again, regarding drug repurposing, in this case for SARS-CoV-2 infection. Now here we have the added complexity of not only the covidome, and I'll show you an example of that in a moment, which comprises the proteins that are targeted by SARS-CoV-2's proteins, the human proteins that are targeted by SARS-CoV-2 proteins. So we have this added interaction of another molecular network, another protein network, interacting with a human disease module, near which are drug target disease modules. So it's a little more complicated in analysis. We began by building the COVID-19 disease module, or covidome, and we did this by identifying from the work of Gordon and colleagues in Lonsdale. This was the peer-reviewed publication of the same work. 332 human proteins, to which 26 of the 27 SARS-CoV-2 proteins bind. We showed that these comprise a clustered network that is discrete and separate from and distinct, statistically, from a random network of the same size in the interactome. And most of these are expressed in the lung. Few are not expressed in the lung and instead are expressed in the vasculature, in the kidney, and in the brain, interestingly. And we used 3 different analytical approaches: the proximity approach I discussed, a network diffusion approach, and a network artificial intelligence approach that was developed by Marinka Zitnik in Zach Kohani's department. And in the interest of time, I won't go through the methods, but if you're interested, you can turn to the paper for more details. We then collaborated with Rob Davey, who heads the National Emerging Infectious Diseases Laboratory over at BU, who has access to the live infective virus and has a high throughput screen that he developed for just such purposes. And we analyzed drugs which we rank ordered based on the analytical approach that, the algorithmic approach that we developed from the strategies in the last slide. And we characterized the drugs as strong, weak, cytotoxic, or no effect. And the difference between strong and cytotoxic is really a difference between those drugs that kill the virus but do not affect the host cell versus those drugs which have an adverse effect on the host cell and may or may not have an adverse effect on the virus. So with this kind of approach, we were able to rank order a host of drugs that were curated from an initial pool of over 7,000. And the key points here are that the drugs that were in clinical trial at the time this analysis was performed are shown in green, and a greater intensity highlights a greater number of trials. So our algorithm really did at least identify those drugs which were, by more conventional experimentation on a one-by-each basis, thought to have an effect or potential effect. Drugs with reported positive effects scattered throughout the literature also appear in the list, like ivermectin. The high throughput screens in vitro virucidal activity yielded a 30% success rate based on our predictions, which is far higher than the success rate of random screenings typically done, which are generally less than 1%. The first screen was done in non-human primate cells, and the second level screen was done in human cells. And here you see that the success rate continued at that high range. And the lead candidates that popped out of this analysis are listed below here, some of which are now being poised for clinical trial through the Active-4 program at the NIH. Now, more recently, in a paper that we put up online but is now in review as well, we looked at the Broad library, which contains drugs that have been approved for human use, but also other drugs which have been in clinical trials whose toxicities are known but not yet approved. And in this analysis, we found many of the same classes of drugs as effective, like prosolaridin, which is a digoxin congener, a cardiac glycoside. But we also found some interesting novel drugs that were quite effective, and one of which is shown here, obatoclax, which is a BCL2 inhibitor used for small cell carcinoma of the lung, and I believe also in patients with lymphoma. And here on the left, you can see in red that compared to remdesivir, which is a positive control, emmetine and obatoclax are quite effective antivirals, as is prosolaridin. And on the right, in an in vivo assay, we explored the effect, in this case specifically of obatoclax, on viral load in the lungs of mice overexpressing the receptor for the spike protein, a human ACE2. And as you can see, obatoclax, regardless of sex, was quite effective at suppressing viral titer. In fact, more so, slightly more so than remdesivir. So this approach, this network-based drug repurposing approach, I think has lots of promise. Obatoclax is an obvious candidate for further drug development for treatment of the disease. Now, a few provisos, a few caveats. We can't tell whether a computationally selected drug will suppress the infection or worsen it a priori, and therefore in vitro high-throughput testing is essential. The list doesn't provide information on other drug targets, which may lead to adverse effects, such as is the case for hydroxychloroquine. And while repurposing approved drugs is assumed to be safe, this is, after all, one of the reasons why repurposing has been aggressively pursued by many groups, since these drugs have already passed toxicity and are used in the wild, if you will. New toxicities may emerge when one treats a novel infection, some novel infection by drug interaction that wasn't known and couldn't have been predicted necessarily. So those are cautions for any drug repurposing exercise. Okay, so in the remaining time, let's turn to food biochemicals in human health and disease, and how network approaches might give us insight into how to develop them, or how to evaluate them. And in an opinion piece that Laszlo and Julia Menichetti and I published last year in this new Nature journal, Nature Food. Yeah, I was surprised too, but Nature Food, this is just highlights the fact that nutrition is on the rise again, as I'll make the case at the end of the talk. We in this opinion piece, we pointed out that there's an enormous amount of, as the physicists would call it, dark matter, nutritional dark matter, as there has been in so-called junk DNA, which we know, of course, has very many powerful effects now and is no longer a black box. The same thing is true when one looks at the chemical components of the food we eat. If one looks at all foods, only about 150 molecules have actually been explored in any rigorous way with regard to their effects in health and disease. But according to large databases like FoodDB, which was developed in Canada in 2 Canadian provinces, Alberta and Saskatchewan, there are now, I think it's up to 28,000 very specific biochemicals that can be identified in this case, the Western food supply. And our notion here is illustrated by this, what's so-called knowledge graph, in which one can put together four putatively linked relationships. One is the food, the biochemical components of food as linked to the food supply. That is, one needs to be able to dissect dietary elements into their substituent components from which the biochemical elements can be identified. Then one can link these two graphs, these two data sets, to protein targets that comprise the interactome, since they directly govern phenotype. And then ultimately, these three sets of associations can be tied to disease, either prevention or treatment or intervention. And that's the basis for the work of Foodome, developing this food omic notion of the biochemical components of food and the relationship to health and disease. Now, to give you a sense of the extent of this dark matter of nutrition, I'm going to show you two foods, garlic and basil, to highlight the fact that the nutritional components as listed by the USDA are far fewer than the chemical components. 150 nutrients, there are over 17,000 chemicals. I think it's it is the foodstuff with the greatest number of biochemicals contained within it. I'm sorry, the nutrient components that are listed in the USDA are 150 and over 17,000 chemical components of all of those foodstuffs exist in FoodDB. In garlic, the USDA lists 67 nutrients, and there are over 2,000 chemical components in garlic, which, as I said, I think is the foodstuff with the largest number of specific chemicals in FoodDB identified. Now, to give you an example of what the USDA leaves out, it doesn't report allicin, the most active ingredient from a biochemical perspective of garlic. It has over 60 biological effects, some of which are listed here. And in the case of basil, one can find a similar limiting number of known species in the USDA database as compared to the FoodDB database. There are over 2,000 chemical compounds, 146 have been quantified, and 2,047 have not, two of which are rosmarinic acid and eugenol, polyphenols, and I'll come back to these in a moment. So how do we move from individual eating pattern to the foodome, to the interactome, to health or disease? Well, a strategy, an algorithm for doing so is listed here. The first point would be to establish what an individual's eating pattern may be, and then to decompose what one eats into dishes from which one can extract the specific food elements and their chemical components. Now, how do you do that? Well, Julia Menichetti, one of our collaborators who works in Laszlo's group, has explored the individual eating patterns of thousands of students and faculty at the University of Milan, where all meals are provided centrally, and one can account for what's eaten by the card swiping that's used to pay for the meals. The next step was to identify recipes for all of those dishes that were eaten, and that can be done with some effort, but not impossibly with internet exploration. So, for example, there are, I think, 25 different recipes for lasagna in different proportions, and one can create a sort of median value for each of the food components, each of the specific food elements of lasagna, and then turn to the FoodDB to identify the chemical compounds in each. And then lastly, one can take a photo, as one can do with some diet apps that give input into what you're eating and keep track of the amounts that you eat. So estimates of the volume of what you eat can be assessed by photos reasonably efficiently and precisely. And therefore, you can develop truly a time activity curve for each of the chemical compounds in each of the foodstuffs that you eat in a standard diet. Now, of course, one of the criticisms would be, well, diets change. And fortunately, thanks to the great work of Walt Willett and his colleagues at the Public Health School, we know lots about how diets change and don't change over time from the food frequency questionnaire analyses they've used in the women's health study. And Walt makes the case that diets really change on average about every decade. And if you think about your own eating habits, I think you might reflect on that point and see that it's true, depending upon how your metabolism changes and how carefully you want to modify your weight, generally preventing it from going up. There are strategies we all use to alter our diet, and those tend to occur in decades. So the goal of this exercise would be to develop this individual foodome that could be barcoded and linked to the individual genome, and specifically gene and protein expression, and their associations with individual diseases through the Interactum that I've reviewed for you. That's the ultimate goal. And in the course of this, we can begin to explore these biochemical components of food as drug. And drug is basically a paradigm for the relationship of, in this case, small molecules to phenotype. And ideally, drugs should either be used to maintain health or reverse adverse phenotypes. Now, here's an example. I think it's the best, earliest example of food biochemicals and their relationship to disease and disease prevention or intervention. And this is from the work at Cleveland Clinic, led by Stan Hazen. It was published in Nature Medicine now back in 2013. And they made the observation that in blood, trimethylamine oxide was found and appeared to be correlated with the risk of coronary atherosclerosis, of atherosclerotic disease. But the curious thing about the observation from a metabolomic perspective is that the precursor to TMAO, TMA, is not a mammalian metabolite. And Stan's group was able to show that it was derived from the metabolic action of gut bacteria from L-carnitine and choline in the diet, red meat. And they showed in a variety of elegant experiments that included both animal models as well as genetic variants to the enzyme that's responsible for oxidizing TMA into its toxic metabolite, TMAO, that this conversion of TMA to TMAO is really important in atherogenesis as it links to the diet. They went on further to show that there are other dietary factors that can beneficially counter the effects of this pathologic metabolic pathway, including allicin from garlic and dimethylbutanol. So this, I think, and Stan has many other stories that are evolving and beginning to be published that make similar kinds of claims in relationship to dietary determinants of disease and dietary interventions or features of the diet, chemical features of the diet, which can prevent or reverse those pathogenic effects. So we took a sort of big data perspective on this notion, and in work we published last year, this is again with Laszlo's group, Sudi Milanoué performed this analysis. She developed this notion of an FWAS, or food-wide association study. And in this case, again, using the data sets from the women's health study in our collaboration with Walt Willett and his colleagues, foods were characterized into beneficial or harmful, shown in green and red, diamonds, and their biochemical nutrient components as beneficial or harmful from the knowledge graphs that we were building. And here you can see the hazard ratio for those with beneficial versus harmful effects over the entire data set. And you can see that there's some species here which are have face validity, provide face validity to the approach. Palmitoleic acid, hydroxyproline, processed meats, all associated with harmful outcomes, whereas white wine, and isoramnitin, for which there are at least a couple of papers as a polyphenol, show beneficial effects. These can also be depicted in a graph, a network way, as we show here, linking both the chemical species and the foodstuffs from which they're derived. Now we'll talk a little bit about polyphenols in the last 5 minutes and give an illustration of how one can identify a specific species that has very specific antithrombotic effects. Now, there were 759 polyphenols. Remember, these are organic chemical structures that are widespread largely in the plant world, that have been known for some time to have largely redox-dependent beneficial effects. They have known protective effects in type 2 diabetes and coronary heart disease. We mapped, and this is Italo Dovale, mapped 65 polyphenols to the human interactome. There were over 19,000 polyphenol disease associations in our knowledge graph. Between 65 polyphenols and the 299 diseases that we studied. Of these, 1,525 were known associations, but over 17,000, almost 18,000, were previously unknown. And here you can see, you perhaps can see the names but not read the targets. The network of targets of each of these polyphenols and their interactions in sub-modules within the interactome. And these include epicatechin ogalate, cinnamonic acid, ellagic acid, resveratrol, genistein. You can see all of your favorite species here. So the importance of this image is that it really illustrates, it reiterates one of the points I made earlier, that synthetic drugs have many targets, and naturally occurring compounds also have many targets. So to focus on a single target is a fool's errand, unless you know that that target really outweighs all other potential targets' actions for the phenotype or disease of interest, which is rarely the case. So we explored this question of whether we could apply the same kind of principles of network proximity to dietary polyphenols as potential drugs. And that point is illustrated here in this complex schematic, where we have a network neighborhood of multiple polyphenol targets adjacent to disease networks, disease modules. The argument or the question here is, could this polyphenol be effective in the treatment of one or the other diseases, depending upon the proximity of its target to that disease module? And in experimental work that Beth Battinelli in our hematology division performed, she explored rosmarinic acid that I mentioned earlier in garlic, and demonstrated that it had significant antiplatelet effects. And we were keyed to that possibility by exploring the network neighborhood of its principal targets, which are Sarc signaling pathway targets that are rich in platelets, such as FIN. And as you can see in her physiological experiments, she showed that rosmarinic acid could significantly inhibit platelet activation as measured by the expression of an activation marker on the platelet surface induced by an endoperoxide congener, for example. And she was also to show that this effect was directly a consequence of altered kinase activity of FIN as a Sarc family kinase. So this is another example. It sort of parallels the potential for developing drugs, developing food biochemicals as drugs that I illustrated in Stan Hazen's work. There are many unresolved issues here that we can spend a long time discussing. The natural foods folks would make the case that the matrix is important, and certainly interactions among biochemicals within the food matrix is also very likely to be important. Here I am railing against reductionism, and yet we focus on a specific species and its effects, largely because that's what we can do. But of course, we're very sensitive to the fact that in a complex system, like the biochemicals that are found together in a food substance, in a foodstuff, we need to be sensitive to and cognizant of potential molecular interactions between those species and their consequences for effects on the interaction network. I'll give you an example from synthetic pharmacology to make that point. This is from the work of Fei-Hsiung Cheng and Laszlo Barabasi, in which they asked the question, if you use drugs in combination, in this case pairs, can you predict, based on the network proximity of their targets, whether those pairs of drugs will be effective or not? And this analysis was done through a retrospective evaluation of large data sets. And they observed that in this case for antihypertensive combinations, if the targets are separated from one another within the hypertension disease module, they're much more likely to have observed antihypertensive effects than if they're close to one another, where in fact there's a greater likelihood of an adverse effect in using the drug combination. So that again raises the issue of trying to dissect the key determinants of a complex foodstuff viewed in combination for optimal beneficial effects. But there's one more proviso here that I have to show you. This is unpublished work from my colleague Vlad Elgart, who's been addressing the question, how does one trace or follow on an individual cell basis the combinations of drugs that may be used in a variety of diseases? Do all drugs get into all cells equivalently effectively? And in what I call his Christmas tree light slide, the answer is most definitely not. Here he labeled 4 different drug species in ways that I don't have time to describe here, with 4 different fluorophores, so that he could monitor by single cell analysis the uptake of each of them. And here you can see they're all different combinations of colors that he was able to discern. And they're laid out here in these contour plots to be more specific in this matrix. So there's and the extent to which the uptake of one drug influences another, how well they correlate or don't, is also an important consideration here. And that point is directly relevant to the question of combinations of nutrient biochemicals and their effects on specific cell functions. So the last slide illustrates what I've tried to summarize for you, that we need to understand, I didn't talk about deep phenotyping in the interest of time and the focus of the talk, but we need to understand individual interaction networks, or reticulotypes as we call them, and their links to specific phenotypes in order to initiate phenotype-specific network-informed drug selection. And I would posit to you today that the food supply is another great source for potential drug selection and drug, if you will, repurposing. And the last point I'll make is that, as I said, nutritional research is on the rise again. In a paper I was privileged to participate in, an opinion piece, a white paper, with lots of folks who know much more about nutrition than I do, led by Daria Mossafarian, who's the dean at the Tufts School of Public Health and a former member of our department here. We published this paper recently, raising a call to arms, if you will, for more investment in research in nutrition, which I suspect will be music to your ears. So I'll thank the people who did the work I mentioned throughout the talk and our collaborators, and be happy to take any questions. Thank you.
Thank you so much, Dr. Loscalzo. That was an amazingly riveting, forward-thinking talk. Very exciting. We have some very interesting questions, and they're flying in here. So I'm going to try and get to a few of them. What do we know about what happens in the human interactome in intermittent fasting practices?
The short answer is nothing. We know nothing about it. The interactome that I showed you is a static interactome. And as the questioner implies, there are lots of dynamic changes to the interactome which could be reflected in interaction. So for example, fasting or exercise, we know can lead to changes in phosphorylation states of certain proteins. And that might affect, either positively or negatively, a particular protein-protein interaction. So looking at dynamic changes in the interactome is a critical additional area of development, which is also in its infancy, but a very important one.
Thank you. Next question is fascinating talk, Dr. Loscalzo. Apologies if this was covered. Is there any effect of heat, cooking on the stability or instability of the interactions and the biochemical properties of foods?
The answer is probably yes. It hasn't been studied. I mean, certainly there are redox changes that occur with cooking, oxidative modification, largely some of which can be quite adverse, and that might in turn affect a nutrient biochemical's interaction with its targets, in some cases beneficially, in some cases adversely. So that really speaks to the issue of preparation. And as the question suggests, we have made no assumptions, or as the question infers, we made no assumptions in our analysis of the diet and its components and the biochemical elements of those components with respect to the preparation of the foodstuff, fresh versus cooked in some way or pan-fried. And those would all lead to different subsets of each of the molecular species because of the likely consequences of heat on some of their functional groups.
It does sound like there are effects of many potential variables on the interactome. One that you looked at in the laboratory studies was sex, and I noticed that luckily you saw the same effect in males and females. But how do you account for a big variable like sex in the interactome?
Part of it's probably a consequence of differential gene expression. So again, here we're just looking at if two proteins are shown to interact, we put them into the interactome. But in other studies, which I didn't have time to go into, in which we look at specific diseases, we explore differential gene expression and then reflect that differential gene expression in a modification of the interactome that's tissue specific, for example. So I think that in the case of sex differences, it's probably a reflection of differential gene expression that's likely to be affected by sex hormones.
And in a related question to that study, it looked like TNF was going down, but nitric oxide also going down, and was that not beneficial for this particular drug?
Right. So in that study, what we showed was that nitric oxide was depressed by TNF, and that with hydroxychloroquine, there was a restoration of some of the nitric oxide production. Not back to normal, but significantly better.
Right. Here's another question. Thank you for the amazing talk. 2 questions. As you know, the stereochemistry of chemical compounds affects their functionalities. How do you consider the stereochemistry in constructing the interaction networks?
Yeah, so these are all L-stereoisomer-based, analyses, and so we haven't really looked at that issue. As you know, that the D-stereoisomers of sugars are the biologically relevant forms in contrast to amino acids. So that really depends upon the species as to which of the stereoisomers you're most interested in, but we haven't explored that here either. These are all great questions. We should put a huge program project together.
Yeah. In your opinion, how reliable are computationally derived databases like FoodDB?
Yeah. We were pretty careful. There are a lot of different food nutritional biochemical databases, and we were careful to choose those in which there were at least objective and rigorous methodologies at play, such as MS, for example, mass spectrometry as a means to measure the species. Now that of course begs the question of the abundance, which isn't always as easily ascertainable by MS methods, although more recent methodologies allow you to do that as well. So some of the other data sets are not at all that rigorous, and the assessment of the content of a species is based on measurements made in other laboratories from curated literature, often not in many of these, there's no guarantee that those observations have been reproduced in a separate laboratory. You know, the same thing holds true for the Interactome. You can find lots of protein-protein interaction data sets online, but you really need to be careful that that each of those is curated in a consistent and objective way. We, for example, when we include reductionist experimental data in our Interactome, we insist that each interaction has been reproduced at least once by a separate laboratory.
Well, we are at time, and I would like to thank you again for a wonderful, wonderful talk. And we'll collect all these questions, and maybe we can discuss them with you at another date.
Of course.
Yeah, thank you so much.
You're welcome. Thanks very
Bye-bye.
much. Bye.